Hello, my name is Jessica Gaines, and I'm a third-year bioengineering student at UCSF. I study how the brain produces commands to control the vocal tract during speech, along with my research advisor, Shri Nagarajan, and in close collaboration with John Hood. And today I'm going to talk a little bit about my project. During speech, the central nervous system works as a control system, sending commands to produce movements in the vocal tract, and also constantly adjusting these commands based on sensory feedback. However, the neural mechanisms that control this process are unknown, as represented by the black box. If we can better understand the processes that are carried out in this black box, then we can understand how the brain controls speech in both typical speakers and in speakers with conditions like stuttering and laryngeal dystonia. This research may eventually help inform treatments for speech disorders like these. One part of understanding how the brain controls the vocal tract is understanding the vocal tract as an object of control. In this project, my co-authors and I explored the output space of a widely used vocal tract model published by Shinji Maeda. This model describes a complex vocal tract configuration using 7 articulatory parameters like jaw position and tongue shape. The model also outputs acoustic data for the sounds that each vocal tract configuration would make, including these 2 frequencies, F1 and F2, that determine the vowel sound. By randomly assigning articulatory parameters, we generated a data set of vocal tract configurations that covers a comprehensive range of vowel sounds. We also added another layer of analysis in which we measured the location and size of the narrowest constriction in both the tongue body and tongue tip regions of each vocal tract configuration. Constriction location and size have a strong impact on the acoustics of the vocal tract, and previous models have suggested that the location of the vocal tract constriction is central to the brain's strategy for controlling the vocal tract. We found that despite the smooth distribution of sounds represented in the dataset, constrictions in the tongue body clustered in these discrete regions: the palatal, velar uvular, and pharyngeal regions. This supports and extends previous research examining constriction locations in speech data. However, since our data set covers the complete range of vowel sounds, we can add that this phenomenon is not limited to any particular language, which supports the idea of a possible biomechanical constraint in the control of the vocal tract, which would have great impact on our understanding of how the brain controls speech. And lastly, I just wanted to acknowledge and thank my mentors, my collaborators, and ARCS for funding this research. I truly appreciate this opportunity provided by the generous ARCS donors. Thank you.